Imagine your body talking to itself, not with, spoken words, but with this incredibly fast, precise language of electrical signals.
Yeah, exactly. Little sparks flying everywhere.
These signals, they allow you to think, to move, to sense the world around you. It's this profound level of speed and complexity hidden just beneath the surface, driving absolutely everything we are and do.
It truly is. When you consider the sheer scale, I mean, the precision of these signals, firing millions of times a second in your brain and, throughout your whole nervous system, it's nothing short of astonishing.
Absolutely. So today, we're going to take a deep dive into the fascinating world of action potentials. These are the fundamental electrical signals that neurons use to communicate.
The basic language, really.
Exactly. You've given us a rich set of sources, and our mission today is to truly go deep. We're talking about mastering this information so you understand it like the most informed professionals in the field, uncovering, every critical concept and surprising detail behind how our own internal electricity works.
Okay, sounds like a plan. To truly appreciate action potentials, though, we first need to understand the neuron's baseline, its starting point. Neurons maintain what's called a resting potential.
Resting potential.
Right. Which is typically around minus 70 millivolts. Think of this as the electrical quiet state, the difference in charge between the inside and the outside of the cell when it's not actively firing.
And how did scientists even figure that out, that a neuron has this baseline electrical charge? It sounds like an incredibly delicate measurement to make.
Oh, it is. It really is. This measurement is achieved by inserting a very sharp, incredibly fine far smaller than a human hair, right through the cell's plasma membrane.
Wow.
It gives us direct, precise insight into the cell's electrical state. And historically, a huge leap in this understanding came from studying the squid giant axon.
Ah, the famous squid axon.
Yes. Its enormous size, up to 0.5 millimeter in diameter, made it an ideal, accessible model for these pioneering electrophysiological experiments. It allowed researchers to actually measure these subtle electrical shifts for the very first time.
Okay, so a neuron has this resting state, this mix of 70 millivolts. But how does it respond to incoming signals? Let's start with small stimuli. How does a nerve membrane, at its most basic level, behave like an electric circuit? Because that sounds like something straight out of an electronics class.
It really does, and the analogy holds up surprisingly well. You can think of the nerve membrane as a parallel RC circuit, R for resistor, C for capacitor.
Okay.
In this analogy, the ion channels embedded in the membrane, there are resistors. They're the pathways through which current, which is really ions, can flow.
Got it. The channels are resistors.
And the lipid bilayer itself, the fatty membrane which separates the electrical charges across the membrane, that acts like a capacitor. It stores electrical charge.
So if you apply a small electrical current pulse to this neuron, this RC circuit neuron, what happens to that current flow?
Right. So initially, when you first apply a current pulse, let's say a small depolarizing or hyperpolarizing one, most of that current rushes to charge up the capacitor part.
Okay.
This causes the membrane voltage to start changing pretty rapidly. But as that capacitor charges up, less current flows into it, and more of it gradually shifts to flow through the resistors, the ion channels.
Ah, so the voltage change slows down then.
Exactly. It makes the rate of voltage change across the membrane slow down, eventually reaching a steady state value.
And the speed at which that voltage changes has a specific name, doesn't it? It's not instantaneous.
Precisely. We call that the time constant, tau. You can think of it as how quickly a neuron's electrical voltage can get up to speed or respond to a change.
Okay.
A neuron with a long time constant will be slower to reach its peak voltage response, while one with a short time constant will respond very rapidly. It's crucial for understanding how fast a neuron can actually react to incoming information.
That makes perfect sense. Now, you mentioned hyperpolarizing pulses making the inside more negative. I remember reading that these show a linear relationship between the current you inject and the voltage change you measure.
Yeah.
Sounds like good old Ohm's law.
It is exactly that. Spot on.
Yeah.
For hyperpolarizing current pulses, once the membrane voltage has stabilized, virtually all the injected current is flowing through the membrane resistance through those channels.
Right.
So if you plot the change in voltage against the amplitude of the current pulse, you see a straightforward linear relationship, just as Ohm's law V equals I times R predicts.
VIR. Okay.
And the slope of that line, the change in voltage divided by the change in current, that's what we call the input resistance, RIN, of the cell. It's a fundamental measure of how much the cell resists electrical current flow.
So if axons are these incredibly long, spatially extended structures, like really long, thin wires in our body, what does this basic passive electrical behavior mean for signal transmission over distance? I imagine that becomes a huge problem.
Yeah. It's a monumental challenge, absolutely. Because axons have significant internal resistance, a voltage change in one region doesn't spread instantaneously or perfectly to other regions. It just fades. Yeah. For these passive responses, the voltage change simply decreases exponentially with distance from the stimulus. This is what we call passive or electrotonic conduction.
And it dies away pretty quickly, doesn't it? I read it's only over a few millimeters.
You're absolutely right. Very quickly. This leads us to the length constant, lambda. It's defined as the distance over which that potential decreases to just 37%, 1 over E of its maximal value.
37%. Not much left.
Not much at all. And for most mammalian axons, typical length constants are only maybe 1 to 3 millimeters.
So, okay, wait. If a neuron signal just fizzles out in a few millimeters, and our axons are sometimes a meter long, like the ones going from your spinal cord all the way down to your big toe, how on earth does a message get from our brain to our toe?
Yeah.
This leaky wire problem is exactly why we need something more, right? This is where it gets really interesting.
Precisely. This passive spread just isn't going to cut it for long distances. To understand this leaky wire problem better, you can think of an axon kind of like a leaky garden hose.
Okay, a leaky hose. I like that.
The plasma membrane is the hose itself, and the cytoplasm inside is the water flowing through. But the membrane isn't a perfect insulator. It has tiny holes, or leaks. These are essentially the ion channels that are always open to some extent.
So current leaks out.
Exactly. The more leaks you have, which means lower membrane resistance, the more water, or current, escapes, and the less actually reaches the end of the hose.
So the efficiency of signal spread depends entirely on how leaky that hose is, relative to how wide it is inside, how easily the water flows down it.
It does. The length constant, lambda, which tells us how far that passive signal will spread, depends on the ratio of the membrane resistance. That's how tight or leaky the hose wall is to the axonal resistance of the axonal cytoplasm. That's how much resistance the water encounters flowing down the inside of the hose.
Right, arm over raw.
A higher ratio of membrane resistance to axial resistance means less current is lost across the membrane per unit length, allowing the signal to spread much further electrotonically, passively.
And axon diameter plays a crucial role here, right? Our sources really highlight how increasing the diameter can dramatically affect that length constant.
Critically, yes, this is key. While membrane resistance, or ATOM, increases linearly with axon diameter, the axial resistance, or ACOM, decreases with the square of the diameter.
Oh, wow. The square.
Yeah. So as an axon gets wider, its internal resistance to current flow drops much, much more rapidly than its membrane resistance increases. The net effect is a significant increase in the length constant, meaning the passive signal spreads much farther down the axon. This is one of nature's simple tricks, let's say, to improve signal spread.
Okay, so that covers the basic passive response, the leaky hose. But what happens when the depolarizing current pulses get larger? That's where things stop being so passive and predictable.
Indeed, that's when things get active. When depolarizing current pulses applied to the membrane increase in size, the voltage response of the membrane starts to deviate from that simple, linear, passive RC circuit behavior we just talked about. You start to see an upward curve, or deviation, in the current voltage plot.
And this deviation tells us that something active is kicking in, something beyond just those passive electrical properties.
That's right. This active change is due to the opening of specific voltage-sensitive NALA plus channels, sodium channels. When the stimulus depolarizes the membrane sufficiently, these specific channels begin to open.
Sodium channels open.
And positively charged sodium ions then rush into the cell. They're driven both by the electrical attraction to the negative inside of the cell and also their concentration gradient. There's way more sodium outside than inside.
So when these sodium channels open, what happens to that depolarization? Does it just get amplified, like turning up the volume?
Exactly. It creates this powerful positive feedback loop. The entry of these positive sodium charges adds to the initial depolarization that was started by the electrode, enhancing the depolarization even further.
Okay.
This is what we call a local response. It involves active changes in the membrane properties, specifically an increase in sodium conductance, how easily sodium can cross the membrane. However, and this is important to emphasize, that while it's a step up from passive spread, it still decreases in amplitude with distance. It fizzles out too.
So it's like a warm-up act, maybe. Not yet the main event.
Good analogy. It's not yet the full self-sustaining solution needed for rapid long-distance communication.
Which brings us to the main event, the suprathreshold response, the all-or-none action potential. This is where the real electrical language of the nerve system emerges, isn't it? The actual signal.
This is it. This is where the true neural language emerges. As those local responses grow with increasing depolarizing current, they eventually hit a critical point. The threshold membrane potential.
The threshold.
This is typically around, say, minus 55 millivolts. Once the membrane potential reaches or exceeds this threshold, a fundamentally different kind of response occurs, the action potential, or sometimes called a spike.
And this is the famous all-or-none principle, isn't it? That it either fires completely or not at all, no half measures.
Precisely. Once that threshold is reached, an action potential is always triggered, and it's a full-sized response. It doesn't matter how much stronger the stimulus is beyond the threshold, the action potential will always have the same amplitude and shape for that particular neuron. If the stimulus is insufficient, subthreshold, it simply fails completely.
Right.
This is a stark contrast to the graded nature of passive and local responses, where the size of the response directly correlates with the stimulus strength. It's like flipping a light switch. It's either fully on or fully off, not dimly lit.
That's a great analogy. Let's talk about the shape of this all-or-none event. Our sources show a typical action potential with several distinct phases. What does that electrical signature actually look like?
Yeah, the action potential has a very characteristic and incredibly rapid electrical signature. First, there's the explosive depolarization, also called the rising phase. The membrane potential rapidly reverses polarity, shooting up from negative inside to positive, overshooting 0 millivolts and heading towards the equilibrium potential for sodium, ENA.
It doesn't quite hit that equilibrium potential for sodium, though, does it? It gets close.
No, it approaches but doesn't quite reach it, typically peaks around plus 30 or plus 40 millibit. This is mainly because the increase in sodium flow isn't infinite, and other channels, like potassium channels, start to play a role.
Okay, so it shoots up, overshoots 0, then what?
Following this rapid rise, we have the equally rapid repolarization or falling phase, where the membrane potential quickly plummets back down towards the resting potential.
Back towards them is at 70 villa.
Right, and often it then briefly overshoots, becoming more negative than the resting potential in what's called the after hyperpolarization, or sometimes the undershoot.
How long does this whole electrifying event typically last? It sounds incredibly fast.
It is incredibly brief. The main phase, from the onset of the rise to the return near resting potential, typically lasts only 1 to 2 milliseconds, just a thousandth or two of a second.
Wow.
The after hyperpolarization can linger a bit longer, anywhere from a few milliseconds to maybe even 100 milliseconds, depending on the specific neuron type.
So to really distill it, what are the 3 key distinctions that make an action potential so fundamentally different from those subthreshold or passive responses we talked about earlier? What makes it special?
Okay, the 3 critical distinctions are, first, it's much larger amplitude, where the cell interior actually becomes positive relative to the outside. That polarity reversal is key.
Okay, big amplitude change.
Second, it's propagated without decrement, meaning it maintains its size and shape as it travels the entire length of the axon because it actively regenerates itself along the way. It doesn't fizzle out.
No decay. Got it.
And third, it's all or none nature. It either fires fully or not at all. Threshold is everything. These are the absolute hallmarks of the neural signal.
This brings us right to the core of the ionic basis of action potentials, the dance of ions. How do these specific ions, sodium and potassium mainly, create such a dramatic electrical event in the span of mere milliseconds?
At its heart, an action potential is a rapid, exquisitely timed sequence of temporary changes in membrane permeability, how easily sodium, Na+, and potassium K+ ions can cross the membrane. Remember, at rest, the membrane is much more permeable to potassium than sodium, keeping the potential near potassium's equilibrium potential, EK, which is quite negative.
Right, around -80 or -90 millirad.
Exactly, but during an action potential, this balance shifts dramatically and continuously. The permeability changes.
So the rising phase, that explosive depolarization we mentioned, that's all about sodium, right? Sodium rushing into the cell.
That's absolutely right. When the membrane depolarizes to threshold, it triggers a rapid, massive increase in the membrane's permeability to sodium. This means many voltage-gated sodium channels quickly snap open. This leads to a rapid influx of positively charged sodium ions, creating a powerful inward sodium current, INA. This current drives the membrane potential explosively towards the positive sodium equilibrium potential, ENA, which is around plus 65 millV.
And that's the positive feedback loop again.
Precisely. It's that powerful, explosive positive feedback loop we hinted at earlier. Sodium current causes more depolarization, which causes even more voltage-gated sodium channels to open, leading to yet more sodium current. It's like pushing the first domino in a very fast, accelerating chain reaction.
So the rising phase is sodium pouring in. What about the falling phase in that after hyperpolarization, the overshoot? How do we get the cell back to its resting negative state so quickly?
Okay, the repolarization and the after hyperpolarization are primarily due to two crucial processes happening almost in sync, but with slightly different timing. First, there's a rapid reduction in that high sodium permeability.
The sodium gates close.
Well, specifically, they inactivate. This is a key point. The voltage-gated sodium channels enter an inactivated state, which stops the influx of sodium. Second, and often starting slightly later but lasting longer, the initial depolarization also causes a slower increase in the membrane's permeability to potassium as voltage-gated potassium channels open.
So sodium flow stops, potassium flow starts.
Potassium flow increases, letting positive K+ ions rush out of the cell down their concentration gradient. So as sodium permeability drops, channels inactivate, and potassium permeability rises, channels open, the net positive charge leaving the cell outweighs any remaining sodium influx. This drives the membrane potential rapidly back down towards potassium's equilibrium potential, EK, causing repolarization.
And the after hyperpolarization, that dip below resting potential.
That occurs if the potassium permeability remains elevated for a period after the main action potential spike. Those potassium channels can be a bit slow to close. This continued outflow of positive K+ ions causes the membrane to temporarily become even more negative than its normal resting potential, before it slowly returns to baseline as those extra potassium channels finally close and potassium permeability normalizes.
This all sounds incredibly intricate, involving specific molecular machines that act as gates, opening and closing with precise timing. Let's delve into ion channels and gates, the molecular machinery itself. What do these channels actually look like, conceptually? How do they work?
Yeah, at the molecular level, these channels are truly remarkable pieces of biological engineering. They're complex proteins. A voltage-gated sodium channel, for instance, is an intricately folded protein or set of proteins that forms a tiny pore right through the cell membrane.
The tunnel through the membrane.
Exactly, a highly selective tunnel. It's built to precisely sense changes in the voltage across the membrane and then respond by changing its shape, opening or closing its pore, or gate, letting only specific ions pass through.
And the gates themselves.
These gates are actually tiny, charged parts of the protein structure itself. Changes in the membrane voltage exert forces on these charged parts, causing them to move, physically opening or blocking the channel pore. This explains those complex, transient shifts in ion flow we see during an action potential.
I found it fascinating how researchers figured out the behavior of individual channels. The patch recording technique sounds like an ingenious way to listen in on a single molecular gate doing its thing.
It truly is ingenious, a real breakthrough. The patch clamp technique, which earned its inventors a Nobel Prize, allowed direct observation of individual channel behavior for the very first time. Imagine a tiny, specially shaped glass microelectrode with the tip diameter smaller than a red blood cell, like incredibly small.
Unbelievably small.
It's gently placed against the cell surface, and then a tiny bit of suction is applied to create a super tight, high-resistance seal between the membrane and the electrode tip. This isolates a tiny patch of membrane, maybe containing just one or a few ion channels.
Wow.
And it allows scientists to record the minuscule electrical current flowing through just those few channels within that tiny patch. It's like putting a microscopic stethoscope right onto a single tiny pore in the cell membrane.
So what does a patch recording tell us about, say, a potassium channel's behavior when the voltage changes?
It shows some really interesting things. It reveals that potassium channels, like many ion channels, seem to spontaneously flip between open and closed states, even at a constant voltage. But critically, with depolarization, when the inside becomes less negative, the channel spends more time in its open state.
So that it's not like a switch that just flips on and stays on.
No, it's more probabilistic. Depolarization increases the probability that the channel will be open at any given moment. What's also observed is that the amount of current flowing through an open potassium channel increases with depolarization. That's because the electrical driving force pushing potassium out becomes greater as the inside of the cell gets less negative or even positive.
And the sodium channel, our sources say, is even more complex. It has that crucial extra gate, the inactivation gate.
Yes, the inactivation gate is the critical difference here, absolutely essential for the action potential's characteristic shape and timing. Like potassium channels, sodium channels have a voltage-sensitive activation gate that opens very rapidly when the cell depolarizes. That lets sodium rush in initially.
Okay, the activation gate opens fast.
But uniquely, they have a second gate, the inactivation gate. With maintained depolarization, this inactivation gate slowly closes, sort of like a plug swinging into the channel pour from the inside.
So it plugs itself up even if the voltage stays high.
Exactly. This means that the sodium current is inherently transient or short-lived. Even if the depolarization continues, the channel will eventually close due to this inactivation mechanism.
So it's like a door with two locks, maybe. One lock, activation, opens quickly when you push the voltage high, but then a second internal lock, inactivation, slowly engages and keeps the door shut, even if you keep the voltage high.
That's a great way to think about it. The sodium channel can exist in essentially three main states: closed, activation gate shut, ready to open, open, activation gate open, inactivation gate open, and inactivated, activation gate might be open, but inactivation gate is shut.
And that inactivated state is key.
It's crucial because once the inactivation gate closes, the channel remains closed and cannot reopen, regardless of voltage, until the membrane fully repolarizes back to near resting potential. Only then does that inactivation gate reset, swinging back out of the way, returning the channel to the closed but ready state. This dual gating mechanism, fast activation, slower inactivation, is precisely what explains the transient nature of the sodium current, allowing the action potential to quickly rise and then just as quickly fall.
Let's bring it all together then. Can you walk us through one more time how this intricate interaction of these two types of gates, the activation and inactivation gates of sodium channels and the slower potassium channels, creates the entire action potential sequence from start to finish?
Okay, absolutely. It's a beautifully orchestrated millisecond by millisecond sequence. First, you get an initial depolarization that reaches threshold. This causes the sodium channel activation gates to open very rapidly, almost simultaneously.
Sodium rushes in.
Right. Massive influx of sodium ions causing that explosive depolarization, the rising phase, the positive feedback loop.
Then the peak.
Then, as the membrane potential peaks near ENA and repolarization begins, two critical things happen almost concurrently, but on slightly different timescales. The sodium channel inactivation gates, which are slower to respond than the activation gates, begin to close, shutting off the sodium influx.
Okay, sodium stops coming in.
At roughly the same time, the voltage-gated potassium channels, which were much slower to open in response to the initial depolarization, now fully open, causing a large efflux outflow of potassium ions.
So one positive current, Na plus N, is shutting off, and another positive current, K plus out, is turning on, effectively making the inside more negative again.
Exactly. As sodium permeability plummets due to inactivation and potassium permeability increases due to channel opening, the net outward flow of positive charge rapidly drives the membrane potential back down towards potassium's equilibrium potential, EK. That's repolarization.
And the after hyperpolarization follows.
Right, because those potassium channels can be slowpokes when it comes to closing. They might stay open a bit longer than needed to just reach resting potential. This continued potassium efflux causes the membrane to temporarily dip below the resting potential, the after hyperpolarization, before they finally close completely, allowing the Ne plus K plus pump and leak channels to return the membrane potential precisely to its baseline, ready for the next signal.
That's incredibly elegant. Before we move on from the channels themselves, I want to quickly highlight some of the incredible molecular details mentioned, like the channel selectivity filter and some really potent channel blockers. It's amazing how precise these tiny structures are.
Oh, definitely. The selectivity filter is just a marvel of atomic precision. It's the narrowest part of the channel pore, and it's exquisitely designed to ensure that only specific ions like sodium or potassium, but not both, can pass through.
How does it do that?
It's based on the precise matching of the ion size, particularly its hydrated radius, that shell of water molecules that naturally surrounds an ion in solution, with the filter's exact geometry and the chemical properties, like the charge distribution of the amino acids lining the pore. It's like a perfectly tailored molecular sieve, only letting the right shape and charge through.
Amazing. And the channel blockers you mentioned are fascinating too, with some pretty serious, real-world, even dangerous implications.
Indeed, tetrodotoxin, or TTX, is one of the most potent neurotoxins known. It's famously found in pufferfish, but also produced by certain bacteria and dinoflagellates involved in red tides. TTX specifically blocks most voltage-gated sodium channels by physically plugging the pore from the outside.
So it just stops action potentials cold.
Completely stops them in affected nerves. Then there's tetraethylmonium, or TEA, plus NO, which is a classic experimental tool that blocks many types of potassium channels. These blockers were absolutely instrumental for researchers trying to tease apart the roles of sodium and potassium currents in the action potential. And sadly, TTX poisoning from improperly prepared pufferfish, fugu, or contaminated shellfish, is a serious clinical issue, causing paralysis and potentially death, precisely because it shuts down this fundamental electrical communication in nerves and muscles.
A stark reminder of how vital these channels are. Okay, so the action potential fires, it travels down the axon, and then it's ready to fire again. But there's a crucial period right after firing where it can't or can only fire with extra difficulty. This brings us to refractory periods and accommodation. What's the absolute refractory period?
The absolute refractory period is the time interval immediately following the initiation of an action potential, during which it is absolutely impossible to elicit a second action potential, no matter how strong the stimulus is.
Impossible. Why?
This is critical for function. It occurs because a large fraction of the voltage-gated sodium channels are stuck in that inactivated state we just discussed. They simply cannot reopen until the membrane fully repolarizes, and those inactivation gates have had time to reset back to the closed but ready state. The critical number of available, ready-to-open sodium channels needed to trigger a new action potential simply cannot be recruited during this time.
So the cell is temporarily offline for sodium channel activity.
Exactly. And then there's the relative refractory period. This sounds like you can fire again, but it's a lot harder.
That's right. The relative refractory period follows the absolute period. During this time, a second action potential can be generated, but only if the stimulus is significantly stronger than the normal threshold stimulus. Why stronger?
Two main reasons contribute. First, some sodium channels are still inactivated, but enough have recovered, reset, from inactivation to potentially start a new spike if the stimulus is big enough. But second, and often more importantly, the potassium channels that open during repolarization are still open or closing slowly. This leads to an elevated potassium conductance, GK.
More potassium flowing out.
Right, which makes the membrane potential more negative or hyperpolarized and generally opposes depolarization. So you need a much stronger depolarizing stimulus to overcome this increased potassium outflow and push the membrane potential back up to threshold. This period often extends through the entire after hyperpolarization phase.
This all suggests a neuron can't just fire indefinitely at super high rates. It has these built-in constraints, these refractory periods, almost like a natural speed limiter or rate limiter.
That's a perfect way to think about it. These refractory periods effectively limit the maximum firing frequency of a neuron. They ensure that spikes are discrete events and travel in one direction, which is vital for how the nervous system encodes information reliably.
What about accommodation? Our sources describe something counterintuitive. What happens if a nerve is depolarized very slowly?
Accommodation. It is fascinating. If a nerve is depolarized very, very slowly, it may actually reach and pass the normal threshold potential without firing an action potential at all.
How can that happen? If it reaches threshold, shouldn't it fire?
You'd think so. But the key is the slowness of the depolarization. It gives the sodium channels enough time to enter that inactivated state before enough of them can open rapidly and synchronously to trigger the regenerative all-or-none spike. The inactivation sort of keeps up with the slow depolarization.
Ah, the inactivation gate closes before the activation gate really gets going properly.
Exactly. Additionally, those slow opening potassium channels also have time to open gradually during the slow depolarization, increasing potassium outflow, which further counteracts the depolarization and makes it even harder for a spike to fire. The cell effectively gets used to or accommodates to the slow, maintained depolarization.
So what happens when this incredibly precise system of channel gates, particularly the inactivation, doesn't work exactly right? Are there real-world clinical consequences?
Absolutely, and some can be quite dramatic, directly illustrating these principles. A classic example is primary periodic hyperkalemic paralysis. This is an inherited disorder where mutations in voltage-gated sodium channels in skeletal muscle lead to faulty or slowed inactivation.
So the sodium channels stay open longer.
They flicker open more readily or stay open longer than they should. This means action potentials in the affected muscle cells last longer, causing increased potassium to leak out of the cells during activity, leading to elevated potassium levels in the blood, hyperkalemia. Initially, this slight buildup of extracellular potassium causes a partial depolarization of the muscle cells, bringing them closer to threshold.
Which would make them easier to excite.
Right, leading to spontaneous, sometimes painful muscle contractions or twitching, myotonia. But, and here's the twist, as the condition progresses or during a severe attack, the sustained depolarization due to high external potassium and faulty channel inactivation becomes more marked. This prolonged depolarization drives more and more sodium channels into the inactivated state, making the muscle cells unable to fire action potentials.
Leading to paralysis.
Exactly. Paralysis occurs because the cells can no longer generate the action potentials needed for contraction. It's a direct, sometimes devastating consequence of subtly altered sodium channel gating, specifically inactivation.
Okay, so we've seen how action potentials are born, how their firing is precisely controlled by these gates and refractory periods. Now let's look at conduction of action potentials, the signal's journey. How does this electrical signal actually travel down the length of an axon without losing its punch, especially considering that problem of passive conduction just dying out over a few millimeters?
This is truly the key to long-distance communication in the nervous system, the solution to the leaky hose problem. As we discussed, electrotonic conduction, that passive spread, is completely insufficient for long axons, which, again, can be over a meter long in humans, hundreds or even thousands of times their length constant.
So it can't be passive.
It can't. The crucial point here is that the action potential is actively propagated. It doesn't just spread like ripples in a pond. It regenerates itself continuously along the nerve fiber, maintaining its full size and shape from the beginning right to the very end.
So it's not just a wave traveling, it's more like a continuous recreation of the wave at each point along the axon, like a line of dominoes falling.
That's a great analogy, exactly. When an action potential fires at one point on the axon membrane, the powerful inward sodium current generated there doesn't just affect that spot, it rapidly flows sideways, both inside and outside the axon, depolarizing the neighboring regions of the membrane.
Okay, local currents.
If this depolarization spreading from the active region reaches threshold in the adjacent, previously resting region, then a new, full-blown action potential is generated right there. This new action potential then generates its own local currents, depolarizing its neighbors further down the axon, triggering another action potential, and so on.
Like lighting a fuse.
Precisely. It's a recurring cycle of depolarization, reaching threshold, and triggering regeneration of the full spike. This allows the signal to propagate down the entire length of the axon while retaining its identical size and shape, ensuring the message arrives at the other end completely intact no matter how far away it is.
That explains how it travels without fading. But why does it only travel in one direction, typically away from the cell body? Because if it's depolarizing areas on both sides of the active region, it seems like it could go backwards towards the start too.
That's an excellent question, and the answer lies elegantly in the refractory period we just talked about. While action potentials often originate near the cell body, at a spot called the axon initial segment, which often has a lower threshold, and they do, in fact, backpropagate from there into the soma and dendrites.
So they can go backwards a bit.
They can invade the cell body and dendrites, yes, which might have roles in synaptic integration. But they do not typically re-invade the part of the axon they just came from and travel backwards down the axon again. Why? Because the region of the axon membrane that has just fired an action potential is temporarily in its absolute refractory period.
The inactivated sodium channels again.
Exactly. Its sodium channels are inactivated and simply cannot respond to the depolarizing current flowing back from the newly fired downstream region. By the time those sodium channels have recovered from inactivation and are ready to open again, the action potential wavefront in that segment has already moved further down the axon. This clever built-in mechanism ensures that the action potential normally travels reliably and unidirectionally away from its origin point, ensuring efficient and orderly signal flow.
That's a perfect example of how what seems like a limitation of the channels, the inactivation and refractory period, actually serves a critical functional purpose in directing traffic. Now let's talk about speed. Action, potential, conduction velocity. Speed factors. What makes some axons lightning fast and others relatively slow?
Several factors influence speed, but a major one, especially in simpler, unmyelinated fibers, is axon diameter.
Bigger is faster.
Generally, yes, for unmyelinated axons. Conduction velocity is roughly proportional to the square root of the axon's diameter. As we discussed earlier when talking about the length constant, increasing the axon diameter causes the internal axial resistance to decrease much faster with the square of the diameter than the membrane resistance changes.
Right, easier flow inside.
This significantly increases the length constant, lambda, meaning those local currents generated by an action potential spread farther down the axon more effectively. This depolarizes adjacent regions to threshold more quickly, thus speeding up the propagation of the action potential wave.
But there's a trade-off with just making axons bigger and bigger, right? You can't just have gigantic axons filling up our entire nervous system. Evolution didn't go that route, mostly.
You're absolutely right. While larger diameter certainly helps speed things up in unmyelinated axons, it also increases the surface area of the plasma membrane. More membrane means increased membrane capacitance.
Why does that matter?
Because it takes more current and more time to charge up and discharge this increased capacitance during each action potential cycle. This effect tends to slow conduction down, partially offsetting the speed gain achieved simply by increasing diameter. So, there's a practical and biological limit to how fast you can make an unmyelinated axon just by making it fatter. The squid giant axon is huge, but it's an exception.
Which perfectly sets us up for the vertebrate solution, the real supercharger for nerve signals. Myelination. This is where things get truly incredible in terms of boosting speed and efficiency in our nervous systems.
Myelination is indeed a groundbreaking evolutionary adaptation, particularly crucial for complex, fast-acting nervous systems like ours. Myelin is essentially a fatty sheath formed by the plasma membranes of specialized glial cells. These are Schwann cells in the peripheral nervous system, nerves outside the brain and spinal cord, or oligodendrocytes or oligodendrolia in the central nervous system, brain and spinal cord.
And they wrap around the axon.
They wrap around the axon multiple times, forming many concentric layers, almost like rolling up a sleeping bag or wrapping electrical tape around a wire. This creates a thick, insulating layer. However, this insulation isn't continuous. It's interrupted at regular intervals by tiny, uninsulated gaps, maybe only 1 to 2 micrometers wide. These crucial gaps are called the nodes of Ranvier.
Nodes of Ranvier. Got it. And that's where the action happens.
Exactly. That's where the action potential really happens in myelinated axons. Because the voltage-gated sodium channels, the machinery needed to generate an action potential, are highly concentrated only at these exposed gaps, the nodes of Ranvier. They're virtually absent from the membrane underneath the myelin sheath, the internodal region.
Okay, so how does this insulation pump these special nodes increase conduction velocity so dramatically? It's like putting a turbocharger on the signal transmission.
It boosts speed in 2 profound ways, fundamentally changing the electrical properties of the axon. First, the multiple layers of myelin act as a superb electrical insulator, dramatically increasing the effective membrane resistance of the internodal segments.
Plugging the leaks in the hose.
Precisely. It's like thoroughly patching up all those leaks in our garden hose analogy, virtually preventing ionic current from leaking out across the insulated segments between the nodes. Second, and just as important, the thick myelin sheath effectively increases the distance between the intracellular and extracellular conductive fluids, acting like moving the plates of a capacitor much farther apart. This significantly decreases the membrane capacitance of the internode.
So high resistance and low capacitance, a perfect electrical combination for speed. Left leakiness, faster charging.
Exactly. High resistances means the current stays inside the axon and travels down it, and low capacitance means the membrane voltage can change much, much more rapidly in response to that current flow.
And because the sodium channels are only at the nodes, the signal actually jumps.
Correct. Because the machinery for action potential regeneration, the voltage-gated Na plus channels, is clustered only at the nodes of Ranvier, action potentials are regenerated only at these nodes, which might be spaced 0.3 to 2 millimeters apart, depending on the axon.
So what happens between the nodes?
The electrical current generated by the action potential at one node flows very rapidly and efficiently through the axon's interior to the next node. This flow is essentially electrotonic or passive, but it's incredibly effective because of the very high resistance and low capacitance of the myelinated inner node segment. It loses very little charge and voltage along the way.
And this is what we call saltatory conduction, from the Latin word saltare, meaning to leap or to jump. The action potential literally appears to jump from node to node.
Yes, exactly that. The action potential appears to leap from one node of Ranvier to the next, skipping over the insulated internodal segments. This mode of propagation is significantly faster orders of magnitude, faster in some cases, than the relatively slow, continuous regeneration that has to occur along every single patch of membrane in an unmyelinated fiber.
This has absolutely profound functional consequences when you compare, say, that giant unmyelinated squid axon to a tiny myelinated mammalian one.
It truly is a profound biological aha moment. Let's take some numbers. An unmyelinated squid giant axon, which is quite large at maybe 500 micrometers, half a millimeter in diameter, conducts action potentials at about 20 meters per second. That's pretty fast, but look at this. A myelinated mammalian axon, which can be as narrow as, say, 10 micrometers, that's 150th diameter, can conduct signals at around 50 meters per second, or even faster for larger myelinated fibers, more than twice as fast with a fraction of the diameter.
So it's not just about raw speed, it's about incredible efficiency and saving precious space within our crowded nervous system.
Precisely. Myelination was a key evolutionary innovation that allowed for the development of our incredibly complex mammalian nervous systems, packed with billions of neurons and trillions of connections, without needing an enormous physical volume. Imagine how gigantic our brains and spinal cords would have to be if all our fast-conducting axons needed to be unmyelinated like the squids. We'd need heads the size of small cars just to fit them.
A mind-boggling thought. And of course, when this amazing myelination goes wrong, there are severe clinical consequences, as we see in demyelinating diseases.
Yes, unfortunately, the importance of myelin is starkly highlighted when it's damaged. In conditions like multiple sclerosis, MS, the immune system mistakenly attacks myelin in the central nervous system, leading to scattered patches of demyelination. This disrupts nerve signal conduction, causing a wide range of neurological problems like loss of motor control, sensory deficits, vision problems, and fatigue.
And it happens in peripheral nerves, too.
Yes. For example, some forms of diabetic neuropathy and other peripheral neuropathies involve damage to the myelin sheath produced by Schwann cells.
What's the mechanism of failure? Why does losing myelin stop the signal?
When myelin is lost, the axon membrane in that region becomes exposed. This drastically reduces the membrane resistance and increases the capacitance in that area. The length constant shortens dramatically. The action potential current generated at the previous node now leaks out easily through the demyelinated segment, and the signal loses amplitude as it tries to spread passively across this damaged region to the next node.
So it might not be strong enough to trigger the next node.
Exactly. If the signal becomes too weak by the time it reaches the next node, or even just further down the demyelinated segment, it may fail to depolarize that membrane to the threshold. This results in conduction block or propagation failure, and the signal simply stops, leading to the observed neurological deficits.
What's also fascinating at the cellular level, according to the sources, is the metabolic efficiency of myelinated axons. It's not just about speed and space.
That's another huge advantage. Myelinated axons are indeed far more metabolically efficient, meaning they use less energy, because the ion currents the flow of sodium and potassium, and therefore the activity of the ion channels and the subsequent work needed by the Na plus K plus pump to restore gradients are restricted almost entirely to the tiny surface area of the nodes of Ranvier.
Instead of occurring all along the axon.
Right, instead of happening across the entire membrane surface like in unmyelinated axons. This means much less Na plus K plus ATPase activity and thus significantly less ATP consumption. Less energy expenditure is needed to maintain the essential ion gradients over time. It's a very energy-saving design marvel, crucial for a metabolically expensive organ like the brain.
And finally, before we leave conduction, in the clinic, there's the concept of a compound action potential. How does that fit into understanding these signals in a practical diagnostic way?
This is a very useful non-invasive technique used in neurophysiology labs in clinical settings. External electrodes, often placed on the skin over a nerve trunk, can be used to record the collective electrical activity of all the axons within that nerve bundle without needing to penetrate individual axons.
So you're recording from the whole nerve at once.
Essentially, yes. Now, most peripheral nerves contain a mixed population of axons, some large and myelinated and very fast, some smaller and myelinated moderately fast, and some thin and unmyelinated, quite slow. When you stimulate the nerve electrically at one point and record further down,
The signals arrive at different times.
exactly. The action potentials in the fastest fibers arrive first, followed by those in progressively slower fibers. So a recording from such a nerve won't show a single sharp synchronous peak like a single axon's action potential. Instead, you see a more complex waveform with a series of peaks or bumps that vary in timing and magnitude, reflecting the summed activity of different groups of axons conducting their signals at different speeds. This complex waveform is called a compound action potential.
And its clinical value.
It has significant clinical value because the shape, size, and timing, latency of the different components of the compound action potential can reveal dysfunction in specific groups of axons. For example, diseases that preferentially affect large myelinated fibers will alter the early, fast components of the compound action potential, helping neurologists diagnose certain types of neuropathies or nerve injuries by indicating which fiber types are primarily affected.
Okay, fantastic. So we've thoroughly covered how these electrical signals, the action potentials, are generated with precision, how they're controlled, and how they're transmitted rapidly and efficiently, especially with myelination. But how do we actually sense anything in the first place? How do external stimuli, light, sound, touch, get translated into this electrical language of action potentials? It all has to start somewhere, right? This brings us to sensory transduction.
That's the critical first step, the bridge between the external physical world and our internal nervous system. Sensory transduction is the fundamental process by which a stimulus, which is ultimately some form of energy like mechanical pressure, light photons, chemical molecules, or temperature changes, is converted by a specialized sensory receptor cell into local electrical signals. These initial electrical signals are typically called receptor potentials or generator potentials.
These receptor potentials, they're kind of like the local responses we talked about earlier. They need to hit a certain threshold to actually send a message onwards to the brain, right?
Exactly. They are typically graded potentials, meaning their size is proportional to the strength of the stimulus. These receptor potentials must be large enough to depolarize the spike-initiating zone of the sensory neuron, or neuron it connects to threshold, thereby changing the firing rate of action potentials in the primary afferent fibers that carry the signal towards the central nervous system. Subthreshold receptor potentials, while they represent detection of a stimulus, don't typically transmit meaningful information centrally.
And different receptors are tuned to different things.
Oh, absolutely. What's also fascinating is the incredible specificity of sensory receptors. Different types of receptors are highly specialized, often exquisitely tuned, to detect only particular types of energy or stimuli. We have mechanoreceptors for touch and sound, photoreceptors for light, chemoreceptors for taste and smell, thermoreceptors for temperature, the list goes on. And interestingly, some animals possess receptors for stimuli that humans cannot sense at all, like the electrical fields detected by some fish or the magnetic fields used for navigation by some birds, highlighting the specialized nature of these biological sensors across the animal kingdom.
Can you give us some quick examples of how these different transduction mechanisms actually work at the cellular level to create that initial electrical signal?
Sure, they vary quite a bit, but here are a few key types. Take a chemoreceptor, like those involved in taste or smell. A specific chemical stimulant molecule binds to a receptor protein molecule embedded in the sensory cell membrane. This binding event then triggers a cascade, often involving intracellular signaling molecules, that ultimately leads to the opening or closing of specific ion channels.
Causing a current flow.
Right, causing an influx or efflux of ions, generating a change in membrane potential, the receptor potential, which is usually a depolarization in these cases. For a mechanoreceptor, say a touch receptor in your skin, a mechanical force like pressure or vibration physically distorts the cell membrane. This distortion can directly pull open specialized mechanically gated ion channels, allowing positive ions like Ne plus or Cap++ to flow in, causing depolarization.
Very direct. And photoreceptors are different again.
Photoreceptors in your retina are a bit unusual and operate quite differently, particularly in vertebrates. Their ion channels, specifically cyclic GMP-gated channels, are actually open in the dark, allowing a steady inward dark current. When a photon of light is absorbed by a photopigment molecule like rhodopsin, it triggers an intracellular signaling cascade involving a G protein, transducin, and an enzyme, phosphodiesterase, that ultimately closes these channels.
So light stops a current.
Yes, light causes the inward current to cease, which leads to the photoreceptor cell hyperpolarizing, becoming more negative. This hyperpolarization is the receptor potential that signals the detection of light. It's a fascinatingly counterintuitive system compared to many other sensory receptors that depolarize.
And the receptor cell itself can vary too. Sometimes the cell that detects the stimulus is the same one that sends the action potential, and sometimes it's a separate cell that talks to the neuron, right?
Yes, there are different arrangements. In the simplest scenario, the receptor ending is just a specialized portion of the primary sensory axon itself. A mechanical stimulus, like stretching a muscle spindle or pressing on a Pacinian corpuscle in your skin, directly distorts the nerve ending's membrane, opening ion channels, causing an inward current, and generating a receptor potential, often called a generator potential in this case. If that potential reaches threshold right there at the first node of Ranvier or the initial segment, action potentials are directly evoked and then travel all the way along that same axon into the CNS.
One cell does it all.
Pretty much. But in many other sensory systems, like hearing, balance, taste, and vision, the transduction process occurs in a specialized receptor cell that is not a neuron itself. For example, in your inner ear, cochlear hair cells transduce sound wave vibrations into electrical signals, receptor potentials. These hair cells then release neurotransmitters onto the terminals of the primary auditory afferent nerve fibers they connect with synaptically. It's this neurotransmitter release that depolarizes the afferent neuron and triggers the action potentials that travel to the brain. So it's often a two-cell or more process. And remember, photoreceptors hyperpolarize, making their synaptic signaling onto the next cells in the retina bipolar cells even more complex, involving modulation of neurotransmitter release.
Okay, so once these signals, these action potentials are generated, carrying information about a stimulus, how does the brain know where in the body or where in our environment that stimulus came from? This concept of receptive fields seems absolutely vital here.
It is absolutely fundamental. The receptive field of any sensory neuron is defined as the specific region of the sensory periphery, like a patch of skin, a region of the retina, or a range of sound frequencies that, when stimulated, affects the electrical activity, usually the firing rate, of that particular neuron.
So for a touch receptor on my fingertip, its receptive field is just that tiny spot of skin it's connected to.
Exactly. For a primary sensory receptor, the receptive field might be quite small and simple, the area it directly innervates. For example, a single sensory receptor on your skin might be activated by indentation of only a very small, well-defined area, and that would constitute its excitatory receptive field. It might also have inhibitory regions around it.
And what about neurons further up in the brain, say in the cortex, that are receiving input from many of these primary receptors? Do their receptive fields get bigger?
Yes, generally they do. Neurons in the central nervous system, CNS, like those in the thalamus or sensory cortex, often have receptive fields that are much larger and sometimes more complex than those of the primary sensory receptors. This is because each central neuron typically receives converging input from many lower-level neurons, effectively summing or integrating information from their individual receptive fields.
So the brain builds up a representation.
It does. And critically, the location of any neuron's receptive field in sensory space is ultimately determined by the location of the sensory transduction apparatus that provides its input, which patch of skin, which part of the retina, etc. This systematic relationship between receptor location and central neuron activation leads to the formation of highly organized neural maps within the CNS. Famous examples include somatotopic maps in the somatosensory cortex, where adjacent parts of the body surface are represented in adjacent cortical areas, the sensory homunculus. Retinotopic maps in the visual system, mapping the visual field onto the cortex. And tonotopic maps in the auditory system, mapping sound frequency. These maps are essential for perceiving the spatial location of stimuli.
Okay, this is incredibly important. We spent a lot of time establishing the all or none nature of action potentials. The fact that they all look essentially the same, just a quick spike. But if they all look the same, how on earth does the nervous system distinguish between, say, a light touch versus a painful poke? Or perceive the difference between a dim light and a bright light? Or hear a quiet sound versus a loud one? This raises a really important question. Given that fixed all or none nature of action potentials, how does the nervous system encode all this rich, complex, graded information about the world around us? This is all about coding of information by action potentials. the neural language?
This is absolutely a fundamental concept, and the solution the nervous system uses is both elegant and remarkably versatile. The answer is not in the shape or the size of the individual action potentials because, as you correctly said, they're stereotyped all or none events. Instead, the nervous system primarily uses variations in the rate or frequency of action potential firing and/or the precise timing or pattern of action potentials as the codes for transmitting information about stimulus intensity, duration, quality, and other features.
Let's start with rate coding. That seems like the most straightforward way. More intense stimulus equals faster firing.
Rate coding is indeed a very common strategy. It simply means that information about a stimulus parameter, like its intensity, is conveyed by the average firing rate of the neuron, typically expressed as the number of action potentials, spikes per second, or hertz. For example, a stronger mechanical stimulus applied to the skin will generally generate a larger receptor potential, which in turn leads to a faster action potential firing rate in the primary afferent sensory neuron connected to it. Many neurons throughout the nervous system employ this strategy, where their sustained firing rate consistently correlates with a specific parameter of a sensory stimulus or a motor command.
What constrains the amount of information that can be encoded just by changing a neuron's firing rate? Are there limits?
Yes, there are definitely constraints. First, there's the neuron's physiological dynamic range of firing rates. The absolute upper limit is set by the absolute refractory period we discussed earlier, meaning neurons can rarely fire much faster than 500, 1000 hertz, and often much less than that sustainably. The lower limit is, of course, zero firing.
Okay, a limited frequency band.
Right. Also, to allow for signaling both increases and decreases in stimulus intensity relative to some baseline, many neurons exhibit a level of baseline spontaneous firing activity, even in the absence of a specific stimulus. Some neurons, like Purkinje cells in the cerebellum, can have very high spontaneous rates, maybe 50, 100 hertz, allowing them to signal changes bidirectionally. Second, the inherent variability or noise in a neuron's firing rate for a given constant stimulus can affect the precision or resolution of this type of coding. The brain often overcomes this by averaging activity across populations of neurons.
Makes sense. And then there's the other main strategy, timing or temporal coding. This sounds more complex, maybe more subtle. Like the brain isn't just counting the beats, but listening to the rhythm or the exact timing of the notes.
It's often more nuanced, yes. Temporal coding refers to situations where the specific timing of individual spikes or the precise patterns of spikes across time or across populations of neurons, rather than just the overall average firing rate over some time window, carries significant information.
You give an example?
One common example that's been studied extensively is the synchronization of spikes across multiple neurons. Neurons responding to related features of a stimulus might fire together in close temporal lockstep, within a few milliseconds of each other. This synchronized firing has been observed in various brain regions, like the visual cortex, and has been linked computationally and experimentally to functions like binding different features of an object together, enhancing signal impact, or directing attention. Other forms of temporal coding might involve the latency of the first spike after a stimulus onset, or complex, reproducible sequences of spikes.
What's the advantage of timing codes over rate codes?
A potential major advantage, particularly for rapid processing, is that temporal codes can often convey information more quickly than rate codes. Rate codes inherently require averaging spikes over some period of time to get a reliable estimate of the rate, which takes time. A precisely timed spike or a synchronous volley of spikes can potentially signal an event almost instantaneously.
Can a neuron use both codes at the same time?
Absolutely, and that's a really important point. Rate coding and temporal coding are not mutually exclusive. Many researchers believe that neural codes are often multiplexed, meaning that both the overall firing rate and the precise timing of spikes can carry different types of information simultaneously over the same neural pathway. This would significantly increase the overall information transmission capacity of the nervous system.
Okay, so bringing all this coding strategy together, when we experience something through our senses, what specific features of the stimuli are being encoded by these patterns and rates of action potentials?
Sensory neurons, using these coding strategies, are remarkably sophisticated in encoding several key features about the world around us. They tell the brain about the precise location of a stimulus, using receptive fields and neural maps as we discussed, its intensity or strength, how long it lasts, its duration, and often information about its onset or offset when it starts and stops, or how quickly it changes.
And the type of sensation itself, touch versus temperature versus light. How is that kept straight? You mentioned the labeled line principle earlier. Can you elaborate on that? It's almost like the brain already knows what kind of message it's receiving, just based on which wire it came in on.
That's precisely the idea behind the labeled line principle.
Hmm.
A sensory modality is simply a fundamental class of sensation like touch, pain, warmth, cold, vision, hearing, smell, taste. Each modality is typically associated with specific sensory receptors that are most sensitive to a particular type of physical energy, the adequate stimulus for that receptor.
Right. Photoreceptors for light, etc.
Exactly. The labeled line principle basically states that the sensory modality perceived when a sensory pathway is stimulated depends entirely on which specific pathway or line is activated, not on how it's activated. Neurons of a specific sensory pathway, say the visual pathway from the retina to the visual cortex, constitute a labeled line for vision. This means that activation of these neurons by any means, whether it's the normal stimulus of light hitting the retina, or even non-specifically by mechanical pressure on your eyeball that makes you see stars, or direct electrical stimulation of the visual cortex during brain surgery, will always result in the perception of that specific sensation, in this case, vision. The brain interprets any activity arriving along that specific neural line as meaning light, regardless of the actual trigger.
That's incredible. It really explains phantom limb sensations too, potentially. Okay, so modality is sorted by labeled lines. How is stimulus intensity encoded in detail, given that action potentials are all or none? It's not like a louder sound makes a bigger action potential spike.
Right. Intensity encoding relies on other clever strategies, mainly exploiting population activity and firing rates. There are three main ways intensity is typically encoded. First, as we've discussed, is the frequency of discharge of individual sensory neurons. Good old rate coding. A stronger stimulus generally evokes a higher firing rate in the active neurons. The relationship isn't always linear, though. Different receptors have different stimulus response functions. Some might be logarithmic, allowing them to cover a huge range of intensities. Some linear, like thermoreceptors over a narrow range. Some accelerating, like some pain receptors.
Okay, firing rate is one way. What else?
Second, intensity can be encoded by the number of receptors activated. A weak stimulus might activate only a few of the most sensitive receptors in an area. A stronger stimulus will physically affect a larger area or volume, recruiting a larger population of sensory receptors and thus activating more primary afferent neurons simultaneously. This greater overall activity converging onto central sensory neurons is interpreted by the brain as a stronger stimulus. Think of it as more voices shouting the same message.
More neurons firing. Makes sense. And the third way?
Third, sensory systems often utilize receptors within the same modality that have different activation thresholds. Some receptors might be incredibly sensitive with very low thresholds, responding even to the faintest intensities of stimulation. Others in the same population might have much higher thresholds, only beginning to fire when the stimulus becomes quite strong. By having this range of thresholds across the receptor population, the nervous system can achieve fine resolution in discriminating intensities over a very wide dynamic range, without requiring any single neuron to have an impossibly wide firing rate range. Low-intensity signals are carried by low-threshold neurons, while high intensities recruit both low and high-threshold neurons.
That's very clever. Okay, so that covers intensity. What about encoding things like the duration of a stimulus or just signaling its start and end? This seems tied to different kinds of receptors that adapt differently over time.
Yes, the temporal dynamics of the stimulus are often encoded by receptors that show different patterns of adaptation. Adaptation refers to the tendency of a receptor and the neuron it drives to decrease its firing rate over time, even if the stimulus is held constant. And what's truly fascinating here is how different types of receptors within the same modality specialize through their adaptation properties to give us very specific kinds of information about a single event.
So some adapt slowly, some quickly?
Exactly. Slowly adapting receptors respond to a prolonged stimulus with a sustained, repetitive discharge. They keep firing, perhaps at a slowly declining rate, for pretty much as long as the stimulus is present. These receptors are well suited to signaling the overall magnitude and the duration of a steady stimulus, like the feeling of constant pressure on your skin.
Okay, they signal it's still here.
Pretty much. In contrast, rapidly adapting receptors, sometimes called phasic receptors, respond vigorously at the onset of a stimulus, but then their firing rate drops very quickly, often ceasing completely, even if the stimulus is maintained. Some also fire a burst only at the offset of the stimulus.
So they only signal changes.
Precisely. These receptors are specialized to signal dynamic changes in the stimulus, like its velocity, how fast it's changing, or its acceleration rather than its steady level. Think about feeling the vibration of your phone or the initial sensation of putting on your clothes. You notice it at first, but the sensation quickly fades due to rapidly adapting receptors. This differential adaptation across receptor types allows the nervous system to extract incredibly rich and precise temporal features about events in the external world, giving us a much more dynamic, responsive, and complete perception than if all receptors behaved the same way.
Wow. So, okay, let's try and wrap this up. We've journeyed all the way from the basic passive electrical properties of a neuron, comparing it to like an RC circuit, right up to the explosive all or none nature of the action potential itself.
Yeah, and we've really dug into the intricate choreographed dance of the sodium and potassium ion channels with their activation and inactivation gates orchestrating that rapid spike.
We understood why the refractory period is absolutely essential for making sure the signal flows in one direction down the axon, preventing chaos. And we marveled at how myelination acts like a supercharger, allowing incredibly fast signal transmission across vast distances in our bodies, enabling complexity.
And we didn't stop there. We also explored how these fundamental electrical events are right at the core of sensory transduction, how we translate the energy of the outside world into this internal language of the nervous system.
And finally, we've truly deepened our understanding, hopefully, of how all the rich information about that world, its intensity, location, duration, modality, is actually encoded, not by changing the shape of the spike, but by modulating its firing rate and its precise timing, carried along specific labeled lines to give us our perception.
If we connect all of this to the bigger picture, it's really quite incredible to consider how these tiny, incredibly precise electrical events happening in mere milliseconds, flickering all over our nervous system constantly, form the absolute foundation of our perception, our thoughts, our memories, and every single movement we make.
You, listening, should now have a really profound understanding of this core language of the nervous system. We aimed for an elite-level grasp of how these essential signals are born, how they travel efficiently, and how they manage to encode everything we experience about ourselves and the world.
Which inevitably raises an important, maybe even profound question to ponder. If the nervous system encodes so much information through the timing and the rate of these stereotyped action potentials, what does that imply for how something like learning and memory might physically manifest in the brain? Could it be through subtle, long-lasting changes in these very firing patterns, perhaps by altering synaptic connections or even the properties of the neurons themselves over time?
That's a fantastic question to leave things on. There's always more to learn, of course, but hopefully this deep dive has given you the solid foundational knowledge, that truly professional-level insight to continue exploring the incredible ongoing complexities of the brain and how it generates our reality from these fundamental electrical whispers.